before introducing our speaker, let me remind you of the events in the coming days. On Friday, Alia Due, who is a postdoctoral fellow here at the center, will be giving a talk in the philosophy of cognitive science, and her talk is entitled Reformulating Rational Analysis. So on Friday here at noon. On Tuesday next week, there is no in-person talk. We have an online lecture as part of the series featured former fellow, where we bring back past fellows of the center to give us lecture. The talk will be online. It will be given by Laura Menati, and the topic is Health and Environment, Understanding the Positive Role of Surroundings, Philosophical and Medical Problems. And you can listen to Laura's talk on Zoom. The link is on our website on the calendar. On Friday, also, while it's not organized by the center, I will put a plug for that talk at 3:30, I guess on the 10th floor, given by... Yes, thank you very much. I was having a senior moment just right there, right now. Thank you very much. So the talk will be at 3:30 again on the 10th floor. Today, it's my great pleasure to introduce Dietrich, who is a professor of History and Philosophy of Science in the Department of History and Philosophy of Science. For many years now, he's been our dear leader, the chair of the Department of History and Philosophy of Science. And he's a historian and philosopher of biology. He needs no introduction, but I will just mention a few things about his work. He's been working mostly on the history and philosophy of 20th century biology with a focus on genetics. He has published widely in this area of research with an interest for outsiders, people on the margins, issues in science involving controversies. So he's published on Richard Goldsmith, for example. He has several books coming out. Maybe we might have an update about some of these books. One of them is A Survival of the Luckiest. It's been accepted, so it should be maybe out in a year from now, 6 months, 8 months. So hopefully the book will be very soon. The title as is right now, Survival of the Luckiest, Perspective on Random Drift in Evolutionary Biology. I've already also mentioned his work on Richard Goldsmith. There's also a book that's under contract in preparation. Richard Goldsmith's Heresy, the biography of a bad reputation. And it's edited also many anthologies and collections, such as a handbook of the historiography of biology. And Dreamers, Visionaries, and Revolutionaries in the Life Sciences, yet another attraction for the margins of science. Today, Mike is going to be talking about something a bit different. Does species diversity matter for biological research? Mike, floor is yours.
Thank you very much. Thank you all for coming. So today I'm going to talk to you about a project that evolved out of a research grant funded by the Australian Research Council on organisms and how they're used in contemporary biology. This research was done with Rachel Ankeny and Sabina Leonelli, and this fine group of people here were sort of intimately involved in what you'll hear about today. This particular project started out with a paper that we wrote in 2014. And this is on publication trends in model organism research in the journal Genetics. And so what we were interested in is just sort of looking at sort of representativeness of how these organisms worked. The article got a lot of attention. So this is an altmetric score, which is measuring Twitter impact, Facebook, and all that kind of stuff. So in the top 5 percentile for articles. But that's the reason why. It's this graph, because it sort of ignited species politics among biologists. In particular, the rat biologists were really obnoxious. And they sort of tweeted this graph out to everybody and said, rats rule. Right? Like, we have the best organism and you don't. And that, yeah, just sort of took off from there. When we got the grant, one of the ideas was to go back at this kind of research, but do it better. My only instructions were to do something quantitative. And so that sort of led to the project I'm going to describe to you today. The sort of more immediate context is this paper by Manfred Lablicher's group at Arizona State. And one of the things that we tried to argue in the original 2014 paper was that model organisms had affected the sort of range of organisms that biologists were using. It actually caused a decline in diversity. Manfred and his group went out and did a sort of mass study of all the research papers they could sort of scrape together easily. And they said there's no impact on diversity whatsoever. So in part today, what I'm going to show you is that this could not be more wrong in both the way it was done and the conclusions that it reaches. So does species diversity matter? The first thing I'm going to do is sort of talk about how we're characterizing biological research, then how we're actually going to measure species diversity in biological research, and then we'll go to three sort of things that I think can explain the trends that we're discovering. The impact of model organisms in contemporary biology, what I'm calling the translational turn, which is a turn toward translation into medical applications, and then single species and the fall of comparative biology as a third possible explainer of these patterns. So this is how we approached biological research. Before, we had just studied the journal Genetics. This time, we decided to pick 8 different major journals that corresponded to major research fields in biology. So cell biology, we picked cell, developmental biology, development. They're all kind of self-explanatory. Not all of the journals went back as far as 1970. We wanted basically a 50-year-ish kind of window so that we can look at trends and change over time. This is just a heat map of the number of articles per journal per year. The significant thing that I sort of want you to stick in your back pocket is the difference between the Journal of Physiology, which sort of never really gets above 100 for real, and then the Journal of Neuroscience, which is up over 1,000. Right? So they can have massively different numbers of articles per year, and that's going to turn out to be significant for our study. But the big assumption, right, the big thing that's informing us is that these journals more or less correspond to disciplinary differences or field differences in biology. Yes, it's not perfect. Yes, we could collect more data, but this is how a rough sketch of sort of dividing the field, how we're characterizing biological research. The next step that we had to do is then to read 25,000 articles. And when the team of undergraduates from Australia did this, they were able to 40,000 species instances. So they literally are going through, they're counting for organisms that are the object of inquiry. Okay, they're not looking for just a mention of an organism. It has to be the thing that's actually being studied. So it's not the kind of thing that you just sort of do a search for and then scrape off every species name. We actually want to know that this is what they're engaging with. In that case, we get 40,000 species instances. It's not 40,000 different species, it's just instances of a species being used in one of these 25,000 articles. So from a database, we would give the students these giant spreadsheets that they would then have to go find the article, read it, and then write down the sort of species, every species that was an object of inquiry. We would then do some data analysis where we'd sort of total these up in different kinds of ways to produce different kinds of quantitative information about the number of species involved in each journal per year. From here, what we did is then get into the business of measuring diversity. The first measure, we're going to use 4 different measures of diversity. They're all borrowed from ecology and evolutionary biology, but they're applied to an ecosystem that is the journal. Okay, so we're not going to nature, we're going to genetics, we're going to neuroscience, things like that. So the first measure is a very simple measure. It's species richness. It's literally just the number of species that you see in a journal per given year. And so this is just the sort of simple tabulation of how many different species appeared in each of those journals. Now, one of the problems here is that you remember neuroscience had thousands of articles per year. Physiology had maybe 100 on a good year, right? And so one of the things that we wanted to do is sort of to take into effect the number of articles that are being published per year and try to normalize those across the different fields. So this is a graph of the number of species if you just make a regular account. If you then normalize the number of species to the numbers of articles, you get a slightly different pattern. They're both log plots, but you see some things starting to stick out. And so if you look at this a little differently, Neuroscience, if you just do the regular count, is actually higher than physiology. If you normalize it, you notice neuroscience is at the bottom and going down. Now, that shouldn't be super surprising because neuroscience should be studying things that have brains and neurons, and most things on the planet don't, right? So we shouldn't expect their diversity of species to be at the top of the chart. Instead, we should expect ecology and evolution to be at the top of the chart. But what we're also looking at here is the trend over time, right? So it's not just that neuroscience is at the bottom of all the other fields, but that it's sharply declining during this time period. And so one of the things we want to understand is that trend as well as the relative position, right? So species richness is the easiest way to do this kind of counting. The second method we used is the Shannon-Weaver Diversity Index. And basically, this is a way of thinking about diversity in terms of how much uncertainty you would have if you tried to select an organism out of a pool. And the difference between species richness and a Shannon index is that this takes into account abundance. So species richness just gives you the number of species. This actually takes into account, well, how many of each species are actually present. So if you had 10 species and you had equal numbers of each, then you would say that's a fairly diverse population. If you had 10 species, but 100 were in species 1 and all the others had a single instance, you might think that's a very lopsided form of diversity. So this takes into account those abundance differences and gives you a slightly different answer. Neuroscience, I'm going to pick on neuroscience all day. They're still at the bottom of the graph, but once again, that's where you expect them to be. But notice up here at the top, right? According to this graph, ecology is actually losing diversity and evolution is gaining species diversity over time, okay? So this one sort of incorporates abundance. The third one is a measure of evenness. And this is just another way of talking about the distribution now across the species categories. So if you have high evenness, then you more or less have equal abundance. If you have one species predominating or taking up a lot of room, then you'll get this sort of literally low evenness. And so you can see in the ecology graph, which is this one here, the dark green, you have this big dip where they lose evenness. So in other words, there's a single species that ends up being highly represented in 1985 that sort of sets it off from the rest, okay? Once again, neuroscience sharply declining, okay? The last measure that we use is a measure of phylogenetic diversity. And phylogenetic diversity is a way of thinking about how far apart you are on the tree of life. And so this is where we got Mark McPeak, who's a biologist friend from Dartmouth. And what he did is he came in and I told him to go into Phylocom and alter the script. And so he went in with R, changed the way the program works so that the program would accept our data about research journals, and then could actually use it to calculate Faith's index of phylogenetic diversity. It's basically plotting all the organisms that are represented in the journal and calculating all of the branch differences to get these scores that you see at the top per year. So the farther apart you are on the tree, the more things on the tree, the higher your score is going to be. And so if you look over here, ecology still at the top, but look at this, molecular biology. That's weird, okay? Molecular biology actually has, for a few years here, the highest phylogenetic diversity of all of these little 8 subdisciplines or fields. That's not what anybody expected, right? They're supposed to be studying molecules, but they have the biggest diversity. So this becomes one of the things that we have to explain. Phylogenetic diversity is also measuring something quite different than the first 3 measures. And so often these results are going to stand apart. You notice here, finally, neuroscience isn't at the dead bottom. Physiology is sort of is fighting in that race for low representativeness on the tree of life. Okay, so we now have 4 measures of diversity, and we can sort of talk now about some general patterns of what's changing in species diversity within biology. So when I plot them all out like this, and I left off all the labels and stuff, because what you just need to sort of see is like, is the line going up, down, or staying the same, or is it kind of a mess? So ecology, very high richness, very high diversity, even though it's trending down a little bit. High evenness, even though you get some little bit of blips in there. And then the phylogenetic diversity is going up. And so in terms of general trends, we have the first three measures that show a declining trend in species diversity, but an increase in phylogenetic diversity. So it could be over here, like what this is saying is that a fewer number of species, their abundance is basically dropping, but they're farther spread out on the tree of life. In other words, they're picking things that are more distantly related to each other than they used to. Evolution shows a declining species richness, but an increasing Shannon diversity, roughly the same evenness, and an increase in phylogenetic diversity. Ecology and evolution, this is actually what we kind of expect. These are usually journals that are dealing with multi-species systems, especially ecology. You notice even evolution score is not nearly as high as ecology score. So that's all, none of that's really surprising. Over here, though, it gets more interesting. So cell biology shows uniform downward trends except for phylogenetic diversity. The same thing with development, but even there, it sort of has a stasis, very low phylogenetic diversity, which means they're all being drawn from the same tree of life, and maybe even a slight decline in the representativeness of species there. When you come over here to genetics, molecular biology, neuroscience, and physiology, molecular biology really stands out as the weird one, right? It's going up in basically every measure except for evenness. And so this is more like what I expected ecology to look like and not molecular biology. I expected molecular biology to kind of blend in with genetics, neuroscience, and physiology to show these sort of declining trends in diversity. So one of the things that we have to explain then is what on earth is going on in the Journal of Molecular Biology that produces these trends. Genetics, this is the thing that we started with in 2014. Turns out to be one of the most boring patterns in the whole thing. We didn't know that when we started out. Much more dramatic sort of patterns of decline in cell neuroscience and physiology. So how do we explain these differences? Our first hypothesis is that it's going to have something to do with the rise of model organisms. And this is in part because Rachel and Sabina are on the project, and this is their thing, right? Talking about model organisms in biology. Now, when we're talking about model organisms, we're talking about this list. This is the list of canonical model organisms that were named by the NIH as part of the Human Genome Project. And we're picking on this list because today, almost everything is called a model organism in some context or another. And it just sort of opens the floodgates to anything being a source for a target system. Here, this is a different category, we want to say. Not only is it more historically significant, there's reasons to think that these might have gotten some forms of preferential funding or preferential consideration by the National Institutes of Health. So it's a different kind of list. And in particular, it's also sort of been the target of some biologists' ire. Some biologists think that investing in this particular list of organisms was a terrible idea, because what it does is it sort of narrows the focus of biologists to a list that is not at all representative of the roughly 11 million species on Earth, right? But the NIH doesn't care about 11 million species. They care about one species, humans, and human biomedicine. So the reason this list is made is not just to buy the love of these biologists, which they're definitely trying to do, but it's because all of those systems are models for human in some capacity or another, okay? So the NIH isn't wanting to increase diversity, right? They want to, if anything, narrow diversity to just humans. But this leads to this sort of hypothesis that some biologists hold that model organism research has created sort of a competitive exclusion situation where it's pushed other organisms out of biological research because there's preference for these model systems. That preference could be sort of the conspiracy theory level, like NIH likes these better than all the others, which they won't ever say in print. But it could also just be that, look, there's really well worked out systems and resources for these particular organisms. More people are working on them. All of the genomes have been sequenced except for Xenopus, the frog down here. And so there's just more to work with. There's more of a community, which is a good reason to jump on these systems. Now, if we see increased model organism use, we should see a decrease in diversity in most cases, right? because what we're talking about here is sort of a reduction to this list of 12 or so species plus humans. And considering that there's 11 million other things to choose from, right? This is sort of a isolation down to a smaller set. So these are graphs that show, these are the non-model organisms used in ecology. The blue lines represent the model organisms used in ecology. It goes from 0 to 100 on each graph. Ecology, not impacted by model organisms, right? Most of the stuff that they're looking at are non-model organisms. Oaks, especially, super popular in this sample. Okay. Evolution, not quite as uniform. There's a few model systems coming in, notably Drosophila and yeast start to come into evolution sort of in this period here, like the late 90s. And so you see a tiny bit of a drop, but that's still not much of an impact at all for model organisms. It changes when we start to get to some other areas. This is molecular biology, still very few model organisms. The black line is human research. So research on either human cell lines, tissue cultures, or directly on humans. And you notice that that line is also increasing. So stick that in your back pocket. We'll talk about that in a minute. Here's genetics. It's a little bit of a mess, but you can see a sharp rise in model organism systems and a fairly consistent pattern or trend of them. And this sort of is the most direct kind of evidence for a replacement, right? It's replacing non-model systems with model systems, but it's doing so back in the 1970s, not in the 1990s when they were named as canonical model organisms by the NIH. And you notice there's still very few human studies in the journal Genetics. There is a journal of human genetics, which, as you would expect, would be about humans. Cell biology, model systems are doing very well, definitely seem to be pushing non-model systems into decline, but you also get this pattern of rising human use. Development, replacement pattern, almost a complete decline of non-model systems in favor of model systems, but very little use of animal or human models. Physiology also shows almost all non-model systems in the beginning and a radical decline as non-model system or model systems rise and human subjects rise. Almost the same pattern or similar pattern with cell for neuroscience, lots of model organisms, non-models going out, humans coming in. So how do we sort of think about these kinds of patterns? In ecology and evolution, there's not going to be any impact from model organisms. Model organisms just aren't in there enough to explain why you have this kind of pattern. Human research also isn't relevant. So there's no real translational impetus to explain these sort of high-level patterns of use. You do still have these drops in species richness, diversity, and evenness in ecology. And so the explanation is not because they all went out and started using model systems. There's just not evidence to support that. So these declines here, and then the positive phylogenetic scores, could be due to a change in comparative practices. In other words, there's a change in the extent to which they use multi-species systems in their research. And that could be one of the things that's explaining these patterns down at the bottom and makes these kind of data about model systems not as relevant to what's happening in those two particular fields. I'll come back to the comparative practices stuff here in a minute. Let's look at molecular biology. Molecular biology is this weird thing. There seems to be no impact from model organisms. If anything, the trend is relatively flat of the blue line down here. There is an increasing use of human systems. We would think that increase in focus on a single species should lead to a decline in diversity, but we do not see any declines in diversity, maybe a little bit in evenness. And we see this very sharp phylogenetic spike. This, I think, can be explained by the rise of microbial species. So molecular biology always focused on E. coli, on Escherichia coli, which is the gut bacteria. Bacteria are lovely to work on because they're small, compliant, and easy to sequence. And so this is really driven by the rise of sequencing of microbial species, of different microbial species, including viruses. And that would explain why the phylogenetic score is the highest. because it includes things like us. So remember, there's all these humans that are actually being studied, and then a whole range of viruses, archaea, and bacteria, which means that the spread on the tree of life is immense, right? And so you get this very high phylogenetic score, even with modest increases in the other kinds of ways of measuring species richness. So molecular biology is an odd one, right? It's odd because they are spending most of their time studying, they're the people that study most bacterial or microbial systems, which is sort of interesting. It's not quite what we expected. Now, genetics. Model organisms here definitely had an impact, but it had a modest impact on diversity, which is what we expect. There's only 12 of them, right? So they're not going to cause a huge rise, and not all of them are represented.
Can you maybe clear a question to remove the Zoom stuff?
Unpleasant.
Yeah, sorry, thank you. Hope so much, yeah.
Yeah, there's nothing new in that one, but yeah.
Yeah, you can get to that one too if you want.
Yeah.
Just go to more and tell it to go away.
Sorry, going to where? More?
Yeah,
More.
hide floating, hide video panel and hide hide floating meter controls about 3/4 of the way down.
Hide floating mute. There we go. Better. Okay. So no real impact from humans on this, and the diversity doesn't really change radically. Notice the phylogenetic diversity is pretty low compared to molecular biology, for instance. And this holds. There's actually not as many microbial systems as you would expect in these systems, even though yeast becomes very prominent in the 1970s. And it is still counted as one of the model organisms. You don't see it making a big impact on the actual diversity scores that we chose to measure at all. Now, I have to click on it because we changed off of that. Okay. All right. Development. Developmental biology shows this declining trend in the first three species scores, and you see the sharp bifurcation as model organisms take over and non-model systems decline. So we think there's a high increase in use in model organisms that's consistent with this decline in diversity. And so here we were more comfortable saying that, look, it's the switch to model systems, in particular to xenopus and to zebrafish, that lead to this sort of decline and this exclusion of other kinds of systems that were used in development, notably amphibians. Amphibians are going into decline, and most people in developmental biology are switching from amphibian-based systems to either mice or to fish or to frogs. And so you see a mass sort of departure, and that's exactly what's reflected in this data. Not a big switch to human-based research at all. Now, cell physiology and neuroscience, I grouped all of these together because they all show roughly sort of similar-ish kind of patterns. Namely, we have a high increase in model organism use, and we expect that to have a sort of negative impact on diversity, which it does in almost every case. Cell biology shows an increase in phylogenetic diversity, meaning that there's now picking things that are farther apart on the tree of life. And that's really meaning E. coli are starting to become a major part of their research program. And so when it goes from humans to E. coli, we always expect that number to go up. So, but notice all of them have sort of increasing levels of model organisms and increasing direct use of human subjects. And both of those patterns should cause these declines in diversity measures, which is what we think is going on. So we'd like to say that in both of these cases, the sort of increased use of model organisms and the increased focus on humans as subjects is leading to a decline in biodiversity. So how do we explain this in more detail? Certainly the increase in model organism research is important. Let me say a little bit more about the translational turn. This is a turn toward human-centric research. It's something that the NIH is very keen to propose. They have translational pipelines, all sorts of things spelled out, because what they really want is to take basic research, apply it positively to human health. That's called the translation process. And they really want to sort of back basic research that they know is going to easily and directly translate into something of medical worth for humans. And so this rise in human-centric research, we think, is reflective of that sort of new emphasis within NIH that comes out of the funding of model organisms, but is in fact more general, right? Because humans are not model organisms. We're separate. The models are supposed to be of us. And so we differentiate here two senses of translation. Narrow sense translation, which is research directly on humans. So humans are the source and the target for these kinds of systems. If that happens, if you get narrow sense translation, research directly on humans, you should see decreases in diversity. And that's exactly what those black lines are showing on the chart. As people start to focus more heavily on humans, you see that trend going up. You tend to see the diversity trend dropping dramatically. There's a wider sense of translation, though, which is research on organisms as sources where humans are the target. This is not necessarily going to lead to a decrease in diversity, because here the humans are the target system, but what we're measuring for diversity are the organismic sources. And to the extent that any organism, not just model organisms, could be sources for human research, then there's no necessary limit on these as sort of influencing diversity or not. Now, remember, the canonical model systems were deliberately picked out because they knew they had human applications, right? And they didn't have to sort of demonstrate that case that any other organism would. Go backward. Oh yeah, so just coming back to those trends. So these black lines, this is the narrow sense translation measure, right? And then the model organisms rising, all contributing to this sort of downward trend. Now, there's a lot that's been written about translation or this kind of phenomenon, even if it's not sort of deliberately used in these ways. So the most famous quote here is from Jacques Monod, the great French Nobel laureate, where he said, "What's true for E. coli is true for the elephant." And this quotation, you're either going to love it or you're going to hate it. Okay? If you're sort of a biologist that appreciates the diversity of nature, you think that this is just an arrogant statement that can't possibly be true. But this statement had a huge appeal for especially people invested in molecular genetics. Molecular genetics, they thought, which they thought was molecular biology, was really getting at the foundational processes that unified all living things. So if you think about the universal genetic code, then yeah, the universal genetic code is shared by E. coli, by elephants, and almost everything else on the planet. Processes of translation, transcription, all that kind of stuff. They seem to be sort of on the trail to discovering all those things. Those are the things that Monod gets his Nobel Prize for, and lots of other people get their Nobel Prizes for, okay? But let's say you're not interested in those basic processes of biology, right? Then you might think that no, absolutely not the case that E. coli is true for the elephant. And you get a quotation like this one down here from 2015. Much like fashion supermodels, E. coli may not be that representative of their close relatives, even their species, and some have never lived in the real world. And this is important. The model organisms and E. coli are highly standardized, very heavily modified systems. They are not natural in the sense that you're going to put out your banana and you're going to collect fruit flies. No, like you right away to the place, you pay your money, you get your specialized stock, even for E. coli, okay? Now, historian Cheryl Logan has tried to explain this sort of rise of a presumption of generality. And she says, prior to 1900, many scientists hoped for great generality, but it was not presumed. Rather, it had to be extracted from the manifest diversity thought to characterize life. Whether general processes could be found amid this diversity was an empirical question. What she's pointing to is almost a process of enumerative induction, right? So you go out and study one organism, find a phenomenon, and then you go find others. So you don't presume that whatever phenomenon you found is automatically shared by everything on the planet. So you would never assume something like Jacques Monod had assumed, right? Instead, you literally have this now inductive program where you try to measure the extent of the spread of the thing. She says by 1929, generality was presumed, and that if you found something exceptional, that was a minor or transient oddity. This date, don't put too much on that, okay? So she's a historian working on the standardization of what's called the Wistar rat, which is the white lab rat. And so she's looking at a group of biologists, and when they're willing to presume that any result they find with the rat is going to be general for everything else on the planet. This date definitely extends into the 1970s for cell biology and developmental biology. Different groups of biologists presume generality at much different times during the history of their discipline. But it's sort of it's an interesting idea, right? That if you really are Jacques Monod, you're not worried about inductive enumeration. You're not worried about anything else. You know that whatever you discover is going to be true for everything on the planet, or at least you think that's the way you think. That's the presumption of generality. But what's really interesting is like, is this idea actually justified, right? Some biologists definitely think it's not justified. And that, moreover, whatever you're studying in the lab has no actual connection to the real world at all. So how do they actually justify even the use of a model system? Here's an argument from Marcel Weber in one of his pieces on philosophy of experiment. And here he's trying to explain the universality of the genetic code. And so he says the genetic code was found to be identical in a number of organisms that are not close relatives. They're not at all close on the phylogenetic tree. So you have a genuinely widely shared trait. He says it's unlikely in the extreme that genetic codes of these phylogenetically remote organisms should agree if they had arisen independently in each of these lineages. So in other words, it's just preposterous to imagine that the universal genetic code has arisen, let's say, 10 million times independently. Instead, it's much more plausible to say all of those organisms have inherited that code from a common ancestor, right? And so here, common ancestry explains the sort of near universality of the genetic code. He calls this an inference to best explanation, where you're contrasting the sort of idea of independent sources of all of these things that by coincidence converge versus descent from a common ancestor as the second explanation, right? This, no one is really, despite Monod, no one is actually making sort of universal claims for any trait, because we know that evolution creates diversity, and it's very rare to find this kind of universal trait. But they are using basically this argument form. And usually it goes something like this. The code for gene X was found to be identical. Identical is in parentheses because the sequences don't actually have to be identical. They have to be close-ish, but not identical. In two organisms from different species. It's unlikely that the code for gene X in the different species would agree if they had arisen independently. Now, sometimes they will, sometimes there's convergent evolution, but the idea is that the way to explain shared features is through common ancestry. So diverse organisms have inherited the gene for X from a common ancestor. Common ancestry explains the shared gene or the shared genetic code. We call these genes orthologous or orthologs of each other. The extent to which genes are orthologous is the extent to which those generalizations will be supported, right? So if you can show that one of your model organism has a bunch of orthologs to humans, then you don't have to study those genes in humans. You can study them in fruit flies. It's the same gene, just in a different system that you can kill at will, raise in masses, just do horrible things to day in and day out that you cannot do to humans. And sure enough, biologists caught on to this. So this is a graph from about a database called OrthoDisease. It's a Swedish database that basically looks for human disease genes and then uses a program called BLAST to search in other genomes for the same genes. And so this graph here is a graph of mostly model organisms. So here's the mouse, here's the cat, here's chickens, zebrafish, flies, C. elegans, and this is chimpanzees up here at the top. And then what you see is the top dark sort of squares here. These are actual disease genes, and it's the proportion that are orthologous or that are shared with humans. So of course, chimpanzees have the highest ratio. The little diamonds that are gray, those are non-disease genes. So sort of general overall genetic similarity between these different species. And what you should really notice here is that the model organisms tend to score not terribly. Mouse is fantastic, right? This is why we can maybe see a trend toward using mice in NIH-funded research, because they're not human, but they have a lot of the same disease genes. And so in fact, that justifies their use as a model system. Some of the things down here, even chickens are not terrible, right? But of course, when you get down here to E. coli, not a lot in common, okay? And the same with some of these things that are way down here on the scale. This one right here, Arabidopsis thaliana, this is the only plant model system. That's very interesting. Why would NIH have picked a mustard weed as a set-in for you, right? Is it because you're sedentary a lot of the day? No, it's because of this orthology. And in fact, there's more articles from the Arabidopsis people who I think have a little bit of a complex about why they're included. But they're really saying, like, we're really doing medical research, like these mustard green seeds, you know, they're really relevant to human cancer, kind of thing. But it's establishing, it's sort of the argument to establish that case for generality, right? For going from source to target, even if the source is something as different as a little weed from you or me. Now, the last sort of thing that I want to talk about is not about model organisms or the rise of sort of human genetics, but what about just single species papers, right? What if all the decline in biodiversity is just the result of people making fewer comparisons between species? What if they're just going to now talk about a single species at a time because it's easier, it's only one of them, there's not a bunch of them. And so this is something that we could measure. And so what we're looking at here are the numbers and proportions of single species papers. And we would expect that if you have an increase in the number of single species papers, you should see a decrease in some of the measures of biological research diversity. Definitely should see a decrease in species richness, because that's just the number of species that are being used. And if you can only use one at a time, we might expect that number to go down. Shannon-Weaver is sort of adding abundance to that. Because the two things are related, we might expect that to go down. The same with evenness. Phylogenetic diversity doesn't necessarily have to go down, because you could still be doing a single species as an E. coli or a bacteria and another one a human and still have that big sort of expansive coverage on the phylogenetic tree. So when we look at the proportion of single species papers, this is sort of the pattern, right? Here's ecology sort of at the bottom. The other way to think about it is that they're still doing the most comparative research of any of the fields, right? They're doing more on multi-species systems, more on ecosystems. But notice even they are going up from around 38 to around 52% towards single species. So there's more single species papers in ecology than there were in 1970. And all of the other lines are showing a similar-ish kind of pattern, except for molecular biology, right? Molecular biology is the only one trending toward multi-species papers. And so maybe another reason why they're showing greater diversity is that they're actually doing more comparative analysis. But once again, because it's easy to do with microbial systems. You can add in lots of different species, and the cost of doing that research is not truly excessive. So the proportion of single species papers could add a little bit to that picture. But it also just raises more questions, like what's going on in ecology that it's moving away from a comparative approach. So let's talk a little bit about this. Ecology and evolution both show an increase in single species papers, but a decline in species richness. That's sort of what we would expect. But the Shannon-Weaver diversity index and the phylogenetic diversity index increase for evolution. So what we can say from this is that the single species papers in evolution are narrowing down to shared species. In other words, there are species that are already represented in the sort of data set. And what they're doing is increasing their abundance. So that's why species richness, the number of species declines, but the Shannon-Weaver diversity index goes up. So they're actually eliminating some species, but the ones that aren't being eliminated are sort of compounding the species that are already in the data set, increases their abundance, which increases the Shannon-Weaver. The shared species are not model organism species, right? But they indicate a species preference even within that literature. So in other words, there's still preferences, even if they're not for model organism preferences, and you still get the sort of maintenance of certain shared species within that data set. And this is a pattern within evolution. there's still a huge number of different species, but within genetics, for instance, one of the shared species that was widely used that wasn't a model organism species was corn, right? Because we like eating corn. And so there was a lot of investment in corn. And so you sort of have a higher Shannon-Weaver diversity index, even as species richness declines because of the presence of those shared species. This is my sort of summary table of where we're at. So remember at the beginning, Manfred's group's claim that there's no decline in organismic diversity. All the negatives are declines in organismic diversity, okay? So the difference here is that we're going sort of journal by journal, standing in for different fields so that we can capture some of those differences. But strikingly, almost all of the fields show some measure of decline in species diversity in the last 50 years. Some of those we think can be very readily explained by the rise of model organism systems, that's cell development and neuroscience. Some in physiology, although it's not as striking. Some of it can definitely be explained by a move toward using direct research on humans. And then that decline in comparative research or preference for single species, we think is actually combining or contributing to almost all of them, except for molecular biology, okay? So contrary to what the ASU group says, we think there is good evidence for a decline in diversity. And we think that some combination of these three factors is at least a good start at explaining what might be going on behind those trends. Now, some conclusions. Diversity has declined. Model organisms have played a role in that decline, but not uniformly across every field of biology. The trend toward human-centered research has also contributed to the decline, especially in those same fields, cell biology, neuroscience, and physiology, but not notably in molecular biology, which is an outlier. It's becoming more diverse, probably because of its focus on microbial systems. Now, a set of meta conclusions about how we do what we do. All of this depends on how we measure research and how we measure diversity, okay? We didn't use the same diversity measures that the ASU group did. I didn't even use the same diversity measures I used in 2014, right? How we characterize research also really matters. We hand curated this set of 25,000 papers. Manfred just scraped whatever he could off of the ASU database. And then throughout the stuff they couldn't actually get a PDF of. It's a different research process to create the data. You can tell that I think our data that's easily acquired but heterogeneous, that's a nice way of saying haphazard, may not be the best basis for any targeted inference, right? So we think that having those poor students read those journal articles for 3 years was worth it, right? Because it gives us a little bit more focus, a little bit more control over that database. That good data is worth the effort. But without more explicit conversations just like this, we think computational HPS is going to remain sort of a place where anything goes, right? Without some shared discussion of standards of good practices and practices that may be less reliable in terms of producing the kind of evidence that we might want. So let me end by thanking the Australian Research Council. Philip Honenberger made our database for us, and these are all the Adelaide undergraduates who spent countless hours reading and enlarging themselves. So thank you very much.
We'll take a few minute break and then we'll continue for questions.